Welcome everyone to our bioinformatics and genomics seminar today. So we're glad to invite Dr. Ryan Yuen from SickKids to give us an interesting talk today. And Dr. Klaus Schmitz from BCH will co-host this seminar with me. And he will introduce our speaker today. So before I turn it over to Klaus, I would like to introduce our very useful RC Wiki page. I think I talk that a lot, almost every seminars. So this RC Wiki page, basically, you can tell what's the upcoming seminars and when was the next seminar and what's the topics, who will give the talk. And we also record all our previous seminars in YouTube. So if you miss any seminars, previous seminars, you can also come to this page. And then if you're interested in anyone, you can click the link. Actually, you can also scroll down. So, and watch the seminar in YouTube at your free time. And you don't have to use VPN. So with that, I will turn it over to Klaus. Thank you, Klaus, for agreeing to introduce our speaker.
Thank you, Ryan. Hi, everyone. Welcome to the genomic seminar at Children's Hospital. My name is Klaus Schmitz. Many of you know me, and I am the co-host and moderator of the occasion. It is an honor for me to present our week guest, Dr. Ryan Yuen. In summary, Dr. Ryan obtained his PhD at the University of British Columbia and continued his interesting genetic career at the Hospital for Sick Children in Toronto, Canada. His name appeared in more than 70 international papers, and he won several prizes and awards. Today, he's going to talk to us to his recent research about a novel computational approach that calls ExpansionHunter Denovo, a computational method for locating known and novel repeat expansion for short read sequencing data. Please, only to remind to the public our simple rules. Keep your distance. Now, just please everyone have off your microphones. Question will be answered at the end of the seminar. Please raise your hand or send us your questions. They are going to be answered in the same order we received them. Our speaker has 45 minutes to present his work. Please, all of us, give the best welcome to our guest, Dr. Ryan Yuen.
Thank you so much for the introduction. And thank you so much for inviting me to this very interesting webinar. So I'm going to share my screen first. Does it work? Can you see it?
Yes, it's working.
Okay, great. It's my pleasure to be able to speak to you about our latest finding on this analysis. So my group have been using whole genome sequencing to study complex genetics in disorders for quite some time already. Although when I say quite some time, I only established my lab 3 years ago. But before that, I did my postdoc and being a research associate in Dr. Stephen Scher's lab in SickKids. And that was the time that when I was working mostly on whole genome sequencing to study the genetics in autism. So this is sort of an extension of what I did back in the day. And so when I got my own position, I started to work on something new that I saw the limitation back then when I worked in as a postdoc. So it really is looking at the tandem repeats in the genomes, and they are also called microsatellites. So this picture in the background basically illustrates this little small tiny microsatellite on the chromosome. And this is the goal of our research, try to map all these tandem repeats and try to see if they are they could be responsible for complex disorders. And as I as I said earlier, because I have been working on autism spectrum disorder, so I begin with applying this to autism spectrum disorder. And my talk will be mostly focused on this aspect. So just a little bit of background about what autism is. We call it autism spectrum disorder because it is a spectrum rather than a definite defined disorder. It is defined by having social communication impairment and restrictive or repetitive behaviors. And right now at 2016, actually, like this is the latest report from the Center for Disease Control and Prevention. It's reported just in US, 1 in 54 children would be affected with autism spectrum disorder. Throughout years of research, we already know that this disorder have a strong genetic basis. And the clinical presentation and the underlying genetics is highly heterogeneous. So what I mean by the strong genetic basis is actually based on the earlier study back in the 90s, when people are starting interested to see what causes autism. And one obvious way to do it would be to compare between monozygotic twins versus dizygotic twins to estimate the heritability. And so as you can see in this graph on the left-hand side, the estimation of concordance rate between monozygotic twins in autism is one of the highest among complex disorders. And it is estimated that the recurrence rates in family would have up to 18 to 19%. On the right-hand side, you can see the concordance rate varies throughout years, from 1977 to more recently 2014. It varies a bit, but the consistent message is that the concordance rate in monozygotic twins is always higher than the dizygotic twins. So estimated the heritability would range around 26% to 93%. Regardless of the changes, one message is really outstanding, which is the strong genetic basis. And so geneticists or people working on autism would be mostly interested to see what are the underlying genetics that could be responsible for the disorder. There are quite a lot of large-scale study done back in the early days with linkage or genome-wide association studies. But most of them are not very successful, and it's mostly because of the heterogeneous nature of the disorder. And so more recently, I would say within this decade of the development of next generation sequencing, people try to and start to use an approach of sequencing trios. More specifically, is sequencing the parents and the child. And in autism, when the child is the only individual that is affected, we call it a simplex family. So the idea was that because that is the first or the only individual affected, so the hypothesis is that most likely there's something that unique found in the child that's supposedly not found in the other individuals in the families. And so when you sequence the trios, you would look for anything that present in the child. In this case, well, obviously we call it de novo variant. So this schematic basically, or this diagram shows an example of the position where there's homozygous A at this position. Well, the child would have a heterozygous mutation of C at the same position. So because it's not present in the parents, will designate it as being de novo. And most often, you will require a lot of this trio sequence and try to identify variants or genes that we currently have variants or mutations, rare variants, or de novo variants, I would say. And you have to compare against another cohort, which is obviously a control, but if it's available, which most of the case in autism spectrum disorder, there is a cohort that is quite widely used, which is coming from the Simon Simplex collection, in which it include also at least one unaffected sibling in the family. So the design of that would be sequencing both the parents, one affected child, and an unaffected child in the family. So a lot of the studies in using next generation sequencing, either exome sequencing or whole genome sequencing right now, would be comparing the rate of de novo variants in the child with autism compared to the unaffected child with no autism in the same family. So using statistical method, there are many ways that you can identify genes or variants that would potentially enriched in the children with autism. And because of the technology and the developed methods, there are a lot of progress in identifying the risk variants or risk genes involved in autism throughout the years. So as you can see in this figure, I also mentioned about the earlier studies in 1975. Beginning that, that was the first evidence that autism spectrum disorder could be genetic, has a genetic basis because of the twin studies. And then the development of technology, including the karyotyping, showing involvement of different chromosomal abnormalities. In this case, mostly would be the large whole chromosome changes. And then there are individual study, cloning of genes, identification of, for example, FMR1, which is responsible for the Fragile X syndrome, but it's also enriched in individual with autism spectrum disorder. And then you can see with the development of microarrays, exome sequencing, and whole genome sequencing, the identification of number of genes involved in autism has been exponentially increased. Some of the notable genes would be SCN2A and SHANK3, for example. You can also see that the prevalence of the prevalence of ASD is also increasing throughout years. And one of the reasons is because of the change in diagnostic criteria using the recommended diagnostic manual. And also the awareness of the disorder as well. So the progress is good. We identify a lot of individuals with potential risk genes or variants, and you can see that the candidate genes scatter across the whole genome. So where are we now? We have identified around 100 strong candidate genes at the moment, including genes and loci. So for example, copy number variations. If we are using microarray for analysis, we can identify around 14% of the individual would have some variants that we know that they are responsible for ASD. And whereas if you have exome sequencing, where they only detect the single nucleotide variants and frame shift mutations or indels, you will find around 16%. So combine the two will be around 20%. And in 2015, there's a paper trying to summarizing this and extrapolate to estimate how many genes or loci would be responsible for autism. And the estimation should be around 200 risk loci and 800 risk genes. And as I mentioned earlier, we need a large number of samples in order to identify the genes. So there was a consortium, which is actually still ongoing, called Autism Sequencing Consortium, which utilized whole exome sequencing to sequence just in 2019, I would say, they sequenced more than 35,000 samples already, of which over almost 12,000 of them would have ASD. So they use a very similar approach where they sequence either trios or quad, and also compare to some singletons where they are either affected with ASD or without ASD. And then using some statistical method, they call it TADA, and also including the PLI score. I'm sure a lot of people familiar with it already, and MPC, which is responsible, very similar to PLI, which is target on exon level for the effect of missense. And with the false discovery of 0.1, they identified around 100 genes responsible for autism. And many of them are actually known before for autism, such as SCN2A that I mentioned earlier, and SHANK3, for example. And through these large-scale studies, we know that many of these genes are responsible for regulation of gene expression, obviously, or neuronal communication. So things are involved in the synaptic functions, and for example, transcriptional regulation. But again, even with all this effort, there's still a majority of the cases still remain genetically unsolved. I'll say more than half of them, even with the current large-scale studies. So the question becomes, where can we find the rest of them? And could there be other ways to identify this loci without only looking at the coding region of our genome? So beyond the coding sequence, you can see that there are quite a lot of repetitive sequence in the genome, obviously, lines, sign. But there's one particular category which is called simple sequence repeats, or sometimes we will call it microsatellite. In this particular study, I will refer them to tandem repeats. And they are more than one base pair DNA motifs responsible for 3 to 6% of the genome, depending on how you define them. Right now, it is, well, when I said it's 40 genetic disorders, right now, actually in these 2 years, at least 10 more diseases or monogenic disorders have been found with expansion of tandem repeats. So this number is increasing, and right now probably more than 50 already. We know that this category of repeats are highly polymorphic and mutable. But more importantly, we know very well about their potential effect on gene expression, which involve potential transcriptional affecting the transcriptional factor binding or epigenetic modification such as a DNA methylation. So if you compare to the protein coding region of the gene, which only encode for 1.5%, you can tell that there are a lot of potential that this repetitive region could contribute to the complex disease disorders. Now, back to the known mechanism or the known genes or loci involved in these repeats. So this review was done in 2018. Trying to characterize around, I think, 20 to 30 genes involved that we know back then, the regulation to the genes and the characteristics. So some prominent example would be, for example, FMR1, which I also mentioned earlier, one of the first genes that known to be responsible for ASD. And the mechanism of regulation of the FMR1 gene with the tandem repeat expansion is quite well known. It is because of the full mutation, which is more than 200 units of the CGG repeats at the 5' UDR region, that causing a silencing of the gene with epigenetic modification. And the other notable example would be the DMPK, which is a gene set responsible for myotonic dystrophy type 1. The mechanism was by RNA dysregulation and causing toxicity. It doesn't. It has no strong link with ASD yet, but there have been reports that report back then that there are some cases with ASD identified with an expansion in the locus within the DMPK, which is the 3' region, 3' untranslated region of the gene. So more about what we know about the functional consequences of the repeat expansion. As I said, in the 5' or the promoter region, it could regulate the epigenetics or transcriptional binding factor, regulating the genes. And if it is in the intronic region, we know that it could potentially impact the RNA splicing, which also leads to the downstream protein function and change in modifications. At the 3' UDR or the intergenic regions, again, it is known to be impacting the stability of the genes or the genome, and then also impacting the RNA function, etc. So now that we know there are potentials in terms of the functions relative to the genes from the tandem repeats, the question is, how can we identify those repeats in a genome-wide level? The concept, if we use the next generation sequencing as a tool, it has a limitation because right now most of the next generation sequencing technology based using short read sequencer. That's right, short read sequence technology, meaning that each of the reads will be short. And if you want to map it back to the repetitive region, it could be challenging, especially when the repeat is expanded. It is because it relies on alignment to the reference genome, and you need specific reads, sequence-specific reads in your reads to map it back in a specific region of the genome. So if it is a repetitive region, it could be challenging. But in 2017, there's a proof of concept where there's a paper published in our collaborator from Illumina. They developed a method called ExpansionHunter, and that is utilizing the repeat and anchor read that's spanning across the repeat. So from BAM file, where when there are parent reads that one-sided, so one end of the repeat, sorry, the read will be sequence specific. You will be able to align that part to a specific region of the genome. And because the other end is attached to the repeat, you'll be able to locate where that repeat is. And this is this method is actually exists even before this paper coming out. But it is really this part when it's the flanking reads and the in-repeat reads that makes a difference, especially the in-repeat reads where the entire reads will be covered by repeats. So the methods include all these repeats and summarize in each locus to estimate the repeat length. And it proved to be quite accurate. But the limitation of that method is that you have to specify the location for the local assembly. And so this is technically not a genome-wide approach, because you have to know where exactly that tandem repeat is located. And most of the time, when there could be some tandem repeat that is not present in the reference genome, then you miss those. And our limitation for the ExpansionHunter is that you also have to specify the motif sequence to the tool in order to estimate the size. So in 2020, working with the same group, we published this work describing the ExpansionHunter Denovo, which do not require specifying the location or the sequence motif. And we just interrogate the entire genome and counting towards the number of in-repeat reads based on the anchored in-repeat reads and paired in-repeat reads. So the anchor in-repeat reads, meaning that just like what I explained, it would be one end where there is a specific reads that you can align to the reference genome, and the paired in-repeat reads would be the repeats that full of repeat. So you don't explicitly knowing where exactly it is, but if you combine that information with anchored in-repeat reads, you have a good estimation. So in this example, if it's within the fragment length of the sequencing, so in this case it's high-seq X sequencing, fragment length would be around 450. If it's within the length, anchor read would get you the maximum, telling you there will be an expansion, but the maximum will be reached to 40 reads. If you include also the in-repeat reads, then you can also estimate size where the expansion is exceptionally larger than the fragment length. Although, as I said, it's just an estimation, but when we try to validate the variance using long-read sequencing, and that's based on one genome, and so we compare the results coming from ExpansionHunter Denovo estimation to the ones that we can be detected in using long-read sequencing. Almost 80% of the loci were validated. So again, it proves that the method works. There are some limitations for this approach. So, which is, for example, you can only detect anything that are larger than the read length. So the read length of the read in, for example, high-seq X would be 150 base pair. So in that case, anything that are smaller than the read length would not be able to detect. So, but the things that are exceptionally large could still be detected. And it can also detect any motif sequence without knowing exactly what the sequence is. And we can apply the same to the whole genome. It's relatively quick. And then we have also proposed a outlier analysis approach where we can define expansion being the ones that are in the outlier region. But the method we describe in this paper relies on statistical method. And in most of the cases, the tandem repeat loci, the size distribution were actually not following normal distribution or any consistent distribution. So statistical method doesn't really apply to all of the cases. So we come up with a new approach of using a non-parametric method to track these outliers. And it's utilizing a computer algorithm, which is called DB-scan. It is density-based clustering algorithm. So instead of statistical outliers, we only flag the ones that are not within a big cluster. So anything that outside we consider to be outliers. And the outliers in the size spectrum, we will define them being expansion. So this is just a summary of our study. In this paper, we have analyzed more than 17,000 whole genome sequencing data, including cohort coming from the Simon simplex collection, which I mentioned earlier, include almost 2,000 families with both parents and affected and unaffected siblings, children. We also have another cohort coming from Autism Speaks whole genome sequencing data from a project called Missing, which is something that I worked on when I was in postdoc in Dr. Stephen Scher's lab. So we utilize this as the replication set as well. Using 1,000 genomes projects, over 2,000 samples, we use this as the frequency set, setting the frequency for each of the locus. And we are mostly interested in the rare cases because of our knowledge before in the genetic architecture of autism. So applying this ExpansionHunter Denovo approach, we identified over 30,000 regions in the genome with tandem repeats. And then we did genome analysis, which I will go through it in more details later. We mapped potential fragile sites, and also using the outlier detection method, as I explained it before, we were able to identify over 2,500 loci that are with rare expansions. Using statistical method, we show that they are highly prevalent in ASD compared to the unaffected siblings. And we also identify some candidate for new loci in ASD. So looking at the distribution of this number of tandem repeats identified per individual, it seems to follow a close to normal distribution. On average, we will have around 2,000 tandem repeats detected per genome. So I say long because this method, as I explained earlier, will only detect any tandem repeat that are larger than 150 base pair. So knowing this, we further characterize what are these tandem repeats that we detected. Out of this over 30,000 repeat containing regions, surprisingly, more than 40% of them have not been previously reported, meaning that they are not present in the database. So even for the known regions where they were categorized as the simple sequence repeat regions, 6% of the ones that overlap with these regions have at least one motif that have not been reported in the reference sequence before. In other words, we actually have missed quite a lot of variability in our genome in this quote-unquote long tandem repeats loci. And on average, each motif would have at least 1.2 different motifs per region. So again, showing how variable in terms of the sequence for this loci. And consistent with other reports, we found that the motifs were predominantly AC or AG rich, depending on the direction or orientation. So, and other interesting features would be that we found that the even number of size of the motif is higher, more common than the ones that are odd number. But that effect only exists before the size of 7 base pair. Some other publications before have been also reported this phenomenon, although I still don't quite know why this is the case. So we further characterized the genomic features of the tandem repeats, and we found that they are enriched in upstream or the 5' UTR regions, but less commonly found in exon or the 3' UTR. So this is expected because the coding sequence is more constrained, so probably less common to find a tandem repeat within the regions. And we also found high GC content and more prevalent of this kind of tandem repeat present in known fragile sites, both common and rare. And so interestingly, if we focus on the ones that are GC rich, we found that there's a list of regions where there have been flagged with a potential low region where there are potential fragile sites but have not been mapped before. And when we looked at one of the latest paper from American Journal of Human Genetics, and I know that the corresponding author also gave a talk to your seminar here as well in this paper. We noticed that the fragile sites that we identified here overlap almost all of the ones that identified in that paper. So again, it emphasized how importance of looking at tandem repeats, and that using even different methods could potentially arrive the same conclusion, where either expansion or methylation of these regions could potentially be fragile sites. Going into more details of what we found, we identified over 23% of the individual with ASD have rare tandem repeat expansions. So in this case, I'm talking about the ones that are outliers in our analysis method. And when we compared it against the children without ASD, which is around 20%, we found that it contributed to an estimate of around 3% of the risk in ASD. And this enrichment is more prominent in the genic region of the genome. More importantly, they are close to exonic region and splicing region. So even for the ones that can be found in the unaffected siblings, you can see that in the percentile, this is the size percentile, which indicates the long length or the large size of the tandem repeats. When we found the ones that are expanded in the proband, even if they are inherited from parents, the size in the parents is actually lower, and so as in the unaffected siblings. So further support what we have seen before for the previously known disease-causing tandem repeat expansion for other disorders. This is just another way of looking at the distance between the expanded or the rare expanded tandem repeats to the location at the transcriptional start site or the splice junction compared to the known tandem repeats or the other expansions. So they are more likely to be found at these transcriptional start site and the splice junctions. And we also compare it against other genes when they are impacting the transcriptional start site or the splice junctions. They seems to be impacting more of the constrained genes, meaning that the genes that would have smaller number of protein truncating variants in the general populations. In terms of function, we identify new functions that is not typically found using exome sequencing or whole genome sequencing. If you recall what I've mentioned earlier, most of the genes that we identified using those methods were impacting the synaptic functions. But in this case, we identified functions that are enriched in nervous system development or cardiovascular and muscles development. So one such example is DMPK, which is something that one of the genes that I mentioned earlier as well, known to be detected also in some cases in ASD. In this case, we confirm one of the cases here using softened blot in the mother, which carry a pre-mutation, 180 units of the tandem repeat. When it's inherited to the child, it has a full mutation of more than 900 units. And the child is also known to us that would have developed myotonic dystrophy as well. So using missing, we use another approach instead of burden analysis. We use a transmission test to see how often the expanded repeat would transmit it to the child for subset of function compared to the rest. So we were able to replicate what we found in the Simon simplex collection where we compare only to the individual with ASD and without ASD within the family. Here, we're simply looking at the transmission within the Simon simplex collection and also within the missing collection. And it's quite reproducible. Combining the two, we also see the same phenomenon where the top hit functions were new nervous system development or cardiovascular muscle development. So in terms of the characteristics, we found that the individual with rare tandem repeat expansions have enriched it in gene sets more in females than males, which is quite similar to what we know in the de novo loss of function mutations or de novo CNVs. And they also have lower IQ and adaptive ability. So all these are very consistent with the loss of function mutations, de novo or rare inherited ones that we found earlier for ASD. Just want to highlight some of the interesting variants or interesting genes that we found. Again, DMPK, and we also were able to validate the FMR1, which in this case, the previous exome sequencing or whole genome sequencing cannot detect. So 2 of them are on X chromosome, 8 of them in autosomal regions. These 2 are the ones that with only 1 motif found, but many of those are actually having RIX motifs found with multiple different sequences. And so this is just another way of looking at how concordance our methods of using ExpansionHunter Denovo to ExpansionHunter. Most of the time we have good correlation, except in some case where there are alternative motifs. So the correlation would be not very good. So this is, again, highlight the limitation of ExpansionHunter. But as long as we know what the target would be, we can actually apply the same way using ExpansionHunter to scan for potential expansion in other disorders. So I just want to highlight some of the examples that I did with other cohorts. We have whole genome sequencing data in over 200 unrelated adults with schizophrenia, and using ExpansionHunter targeting on specific known regions, we were able to identify some large expansion in, again, the DMPK and other known disease-causing region. Similar in epilepsy that we identified. So all these are unpublished data. We also identified a expansion in DIP2B, which is a known expansion that responsible for mental intellectual disability. So in summary, I just want to highlight some of the key points that I highlighted in the talk. This pipeline that we developed were able to accurately detect repeat expansions from short-read whole genome sequencing data. And we now know that the tandem repeats are highly variable across different individuals in the genome. And the repeat can potentially contribute to ASD and also hopefully address some of the missing heritability in other complex disorders as well. So with that, I'd like to thank my team members in my lab and also the collaborators. This is a big collaboration with a lot of genome data. So it cannot be happen without all the collaborations. And so with that, I would like to conclude and take your questions. Thank you.
Thank you so much, Ryan. And I'm not sure how many questions you have, Klaus, but I have one question from the audience.
Yes, we can start with the question in the audience, please.
Yeah, so Laurie, can you actually unmute yourself and ask the question?
Yeah, thank you, Ryan, for speaking. So my question was, when you compared your results of ExpansionHunter Denovo readings to long sequence reads, were both sequencing methods done on families or on single individuals? And a follow-up is, do you think sequencing families or single individuals has an impact on detecting tandem repeats?
So the first question is that the correlation is with individuals, so not by families.
Okay.
For your second question, so in terms of detection, whether or not having families doesn't matter. So whether or not having family members doesn't matter. What matters is the analysis. So if you try to identify novel loci where there hasn't been related to any disease before, especially in complex disorders. In our experience, for example, in autism, you need to have a large number of genomes in order to have a statistical power to pinpoint individual loci being relevant. An alternative approach is to sequence in trios or with family members. So I've also shown where you can look at the transmission. These are two different ways of potentially addressing the discovery of new tandem repeat loci for disorders. But again, both of the methods require large scale of genomes. So that's I think one of the take home message in this paper or this talk.
Okay, thank you.
Anyone, any else have any question? I have a very specific question. If you go to your slide 18, maybe you can share your screen.
Okay. Let me go back to that
slide 18. Yeah.
Right.
Yes, you say that more than 50% are rare repeat expansion. So which database you use to filter or to calculate this allele frequency? Because we know very little about repeat expansion. So I don't believe there are trustful database about it, but
right.
Maybe I am wrong.
Yeah, so this may be a bit confusing. So this slide, I'm not talking about repeat expansion. This is something just anything that we were able to detect using ExpansionHunter Denovo, which means that there are more than 150 base pair. So we define expansion only when they are outliers. So these are not outliers. These are just anything that we were able to detect using ExpansionHunter Denovo. So what it means here is that out of the 30,000 regions where we identified, almost half of them have not been previously reported. Now, what has been reported before would be around 1 million regions in our genome they call it simple sequence repeat. And so this is just another way of describing tandem repeats. So the overlap is any overlap. So if the regions that we identified would overlap any of the regions that they previously report. And so, which means that around 15,000 of them have not been reported in those databases before.
And okay, yeah, it sounds like we need more. Why you didn't use your control data, like your unaffected parents or unaffected siblings? Why you use different? Yeah, wondering.
So when you say our own, you mean the missing data?
No, well, you have trios, so you have the father and the mother, and they are unaffected. So why you didn't use that as to calculate the allele, to create your own database to filter?
So the statistical method that we use, we have to have controls, right? So the control is the unaffected siblings. So in a sequence, like in the family, we have sequence. Actually, we didn't sequence. We just get the data from the Simon Simplex collection, where they have two parents, one affected child, and one unaffected child. So we use the affected child versus the unaffected child for the statistical method. Mm-hmm. But in order to know what is rare, we need to have some other basis. And so we use the 1,000 genomes projects data to set the frequency, just like what we typically do with exome sequencing or whole genome sequencing. So there's a nomad that you can check the frequency, right? So that's the frequency that you will check, instead of using the internal frequency, because we want to do the statistical comparison there unbiasedly.
Yeah.
Because otherwise, if you want to remove anything rare within your family, you will reduce your power already, because a lot of the rare one would actually be involved, right?
Yeah, and then you're talking about Denovo, so they should not be this rare in the parents, but maybe other families have it, or maybe the father or something. We have a question from Ben Weisberg about QC. Maybe Lou, you can unmute him and introduce yourself, please.
Yes, so I'm Ben Weisberg. I'm in the TGG group at the Broad Institute, and yeah, I really enjoyed that. I have a sort of general question first about whether QC, whether there were any sort of non-obvious QC steps that you had to do after running ExpansionHunter Denovo. So you mentioned that the sort of concordance with ExpansionHunter was around 80%. Like, are there things about samples that you found to sort of affect the quality of the ExpansionHunter Denovo calls?
Very good question.
Yeah, it is a very important step, and we did a lot of work on that. And so if you, well, I will recommend you read the paper for sure. So, but then there are some obvious things that needs to be aware of is the library prep. So whether or not they are PCR or PCR free would make a difference based on our analysis. Whether they are cell line or not, and then whether they are sequenced in the same platform. So different platforms using different read length would create different threshold. As you can see, if you remember what I said, ExpansionHunter Denovo only detect anything that are longer than the read length. So if the sequence you use with different read length, it is incomparable because they would have different number that they were able to detect. And then we also, so you saw that I plot the distribution of the number of tandem repeat identified per individual. So we took out the ones that with exceptionally high number of tandem repeats. And we actually we are not sure if those are false call or genuinely biological, having more expansion, more tandem repeats. But to try to avoid the bias, we just remove the outliers in terms of the size, like the number of tandem repeat detected. So we did quite a lot of QC already. And back to the point where you said around 80% validation rate, it was not compared against the ExpansionHunter. It was compared to the long read sequencer, where the long read sequencing would able to potentially detect all the repetitive regions.
Okay. Sorry, did I answer your question, Ben?
Yes, thanks.
Okay. Unfortunately, we need to. Sorry.
I have a question.
Yeah, please.
Yes. So basically, I have two quick questions.
So let me say that I really enjoy your talk. I have to leave. I have another meeting. Nice to meet you. And I hope I see you at Children's Hospital someday personally. Thank you very much.
All right. See you, class.
See you. Thank you, Lenny.
So two quick questions. The first question is that I know for this tool, ExpansionHunter is mainly for the whole genome sequence. So I was just wondering, is that also can be used for the whole exome sequence also?
That's an interesting question. We haven't really used exome sequencing, but in theory, it should not work for exome sequencing because exome sequencing is captured based. There are two reasons that it would not work. One, the capture base would have bias on the capturing. So similar to PCR approach, where it will generate some potential false expansions. That's one. The second one is that what we identified so far, most of the ones that contribute to disorders are in the intronic regions. So if we use the exome sequencing data, we would not be able to detect most of the ones that we've signaled in our data set here.
Okay, got it. That makes sense. And the second question is that for this software, is also okay to detect the zygosity?
Well,
that seems like difficult.
It is not. It cannot. So there are quite a few limitations in this tool where we don't know the orientation, we don't know the zygosity, we don't know the precise number of units. So if you read the paper, most of the time when we talk about the size, we only use the percentile or the normalized length. So we, if we want to know the exact size or the zygosity, we would use the ExpansionHunter. So knowing where exactly the region is, and then knowing in that particular region what the detected motif is, then we can size the repeat more precisely. But that is also a bit challenging. Probably I don't have time to elaborate, but still proven to be challenging.
Got it. All right, I think that we don't have any more questions with that. Thank you so much, Ryan, for the interesting talk.
Thank you so much.
And also we have a lot of seminars on November 3rd. The topic is about RNA splicing. Thank you, everyone, and I'll see you next time.
Thank you.
Thank you again. Bye, Ryan.
Bye.